This is our VDL staff. We got 22 faculty, 100 technical staff, 20 graduate students and residents, and if you look over here in this corner, you know, there's the proud daddy of that group, Roger Main, and Roger's done a tremendous job for us in the industry, and so that's his team. A little bit about our diagnostic lab, and I think what I say about Iowa State certainly holds true for the other NOM labs that work with the pig industry, but we're the only full-service and fully accredited lab in Iowa, of course, and we certainly spend a lot of our time working with the pig industry, not just in Iowa, but across the United States. We probably do more diagnostics, pig diagnostics, than any other lab in the world. And this is, you can see the influence of PED. 75% of our cases are swine that come to our diagnostic lab, 10% bovine, 10% poultry, and then 5% dogs, cats, and everything else. And if you look at this, you can see the porcine graph is on the top, and this is actually revenue. And so you can see that PED has not been a bad thing for our diagnostic lab, although that's certainly not the way Roger or we look at it. We're trying to help the industry. But that's a pretty good synopsis of what we do. I think Lisa mentioned the 29 states now. Lisa, is that correct, with Vermont? But of course, when you look at where the pigs are in the United States, and then you, for the media that are in the room, when you look at where the cases are, and now Vermont up next to New York, it pretty much mimics where the pigs are, is where the cases have been, or the majority of the cases. And so just a little review on the coronaviruses in general. What we have in the United States, of course, TGE is the one that I had from my age group had most experience with in my past as a practitioner. Now we got the PEDv, the porcine delta coronavirus. Lisa talked about a little bit, but one thing, you know, field reports suggest that it is a pathogen similar to TGE or PED, although the viruses are not related. But we haven't gotten around to putting this in pigs yet to really understand the pathogenicity of this virus. It's just something that we're finding out there in the course of looking at all these different cases, almost 5,000 cases in the various NOPS lab. The other one that we still find in test for on occasions, especially with export situations, is the respiratory coronavirus. That virus came into the United States in the early days, my practice days, somewhere in the mid-'80s. And actually, it seems that even though we tried to turn it into a disease pathogen, really it's been a benefit to us because it's virtually eliminated the pathogenicity of TGE virus, which is still around, but it has been more of a friend than a foe. So coronavirus diagnostic specimens for TGE, PED, and then now the deltas, intestine, feces, fecal swabs, oral fluids, very effective at finding PED by PCR. Of course, it's contamination, environmental contamination, but pigs test their environment, and then when they use the ropes, we can pick that PED virus by PCR up. And then, of course, we have the serum antibody test. Several of the labs have that. For us, with our antibody testing, right away, once we were able to culture the virus, and the virus can be cultured on Vera cells and sometimes Mark cells, but the virus is very difficult to grow, and so it took several weeks in our diagnostic lab to develop some of these cell-adapted strains so we could get around to making our antibody test. And the IFA was the first one that was developed at Iowa State and still in use primarily. It's our primary antibody tool at Iowa State right now. The ELISA, University of Minnesota, I think is routinely running their ELISA for PED virus. We have one that we finished validating. We'll have it up and going probably next week at Iowa State, Ohio State, and South Dakota State. I think, are working on ELISA tests. The commercial ELISA tests from Asia are pretty much, the sensitivity and specificity is extremely bad in both cases. So we have not been able to use commercial ELISA tests that have come in from Asia. And then we have the TGEV respiratory corona differential test that we still run primarily for exports to prove that it's respiratory corona and not TGE virus. And then from a serology standpoint, we don't have any tests available for the Delta coronas yet, although that's being worked on. So a little bit about the IFA test. I don't know if you can see it, but the disadvantage of the IFA is it's labor intensive and it's technician dependent. You have to have a well-trained technician to read these tests. You can get some background fluorescence. It can be confusing on some occasions. Those of us that remember the old IFA for PRRS virus, we were fooled, occasionally by those results. Were confusing, but we think we've got a really good IFA test at Iowa State, and we do the screening assay at a 1 to 40 dilution. So if it's less than 1 to 40, those are called negative. Greater than 1 to 40 are positive. But we also can do a 4-fold dilution test and give you some idea of what the titer, how much virus is there, how much antibody is there against the virus. What we don't know is that the IFA results don't seem to relate to immunity necessarily. So, I mean, you can't feel good when you get high IFA titers on a group of gilts you're going to introduce into your pig, into your herd. You don't really know if those animals are still shedding the virus or not based on IFA. So you can't overinterpret those tests. IFA immunity, the antibody is only present for fairly short time, so it can also be deceiving. Animals that were exposed 6 months ago or 7 months ago may be negative on the IFA test by the time you test them. So here's just the antibody curve. This is based on Dr. Madsen's work with pigs, and you can see that that 1 to 40 cutoff somewhere out here around 60, 62 days, those animals dropping, and even though they're recovered, they're negative on IFA tests. And you can see serum neutralization, however you interpret that, we see a pretty good drop in neutralizing antibodies as well in growing pigs. So PED and TGE virus diagnostics, we are doing immunohistochemistry for PED virus. I think what happens, though, this virus is so fast, replicates so quickly in the gut endothelial cells. By 36 hours, the young pigs, baby pigs, pretty much have washed everything out. Their gut is pretty much nude, and so by the time you run the immunohistochemistry, you can see there's 36 hours. There's just very little virus lights up on that immunohistochemistry at that time. So respiratory, the Delta coronas, we've experimented with the IHC immunohistochemistry, and so far we don't have much success. So coronavirus molecular diagnostic assays, I think since mid-May, when we diagnosed the first case, actually it was mid-April, that case was submitted, I think, April 14th. It took them, Greg Stevenson actually was the guy that figured it out that this is probably PED virus in that first week or two after we got that case. And then KJ Yoon, Dr. Yoon, did a whole genome sequence on the virus that got out of that case. And we knew it was, we thought it was PED virus, and we had to send it over to NBSL for confirmation. And it was about the 1st of May that they confirmed that the virus was present. But since that time, if you look at these, everything in red are assays that we've developed in our diagnostic lab since the 1st of May. And you can see the PED virus PCR. Initially, we were doing 2. We've got a PED differential PCR, which can distinguish between the original isolated strain and this strain that I'll talk about in a minute that we're not sure if it came in together, but certainly they came in fairly close to each other in April, May, or June. Then we've got the Delta Corona PCR up and going. The pan corona actually helped us understand that this was PEDv virus. Dr. Yoon had that pan corona, which will detect all coronaviruses that are known. And so they ran that test, and they knew that they had a coronavirus, but all the TGE tests were negative. And then they sent the samples out to other labs, and they confirmed that it's not TGE, even though it looks like TGE, but we knew it was a coronavirus, and so that led us down the path, helped us understand what we had. But we're doing more and more of the PED virus sequencing now, and that's based on the spike, about half of the spike gene, and then, of course, now we're also doing the Delta coronavirus sequencing at the lab. And so here's a PED PCR testing at ISU. You can see since the break began, we've done about 15,000 PCR tests for PED virus at the diagnostic lab. And this is just everybody's seen this, I think, but this is the case, the number of cases, and then based on whether it's unknown grow finish or sow, and a lot of them are unknown because we just don't know what the source was. So, and that's the cumulative cases by farm type, and it's been a steady rise. It does look like that the number of cases peaked back maybe a week or two ago. It's still fluctuating, but it looks like we may have reached a peak, and hopefully warmer summer weather will help us a little bit with this virus, because it is an envelope virus and it is susceptible to UV light and drying and heat. So at our lab, the PCR is a nucleoprotein, or the matrix gene. We think the tests are, of course, very sensitive and specific. We've got same-day testing. Samples come in, the PCRs run that day. Samples that we're testing are intestine, feces, fecal swabs, oral fluids, and then also we've had quite a few environmental samples come in for PCR testing as well, and I think I've got that in another slide. For the sequencing, the spike protein is what we're sequencing, and it's 2.2 kilobases. It's about, I think, about half of that spike gene is what the sequence is based on. The matrix and the nucleoprotein is highly conserved in these viruses, but the spike gene is variable. So this helps us understand, is this virus, which virus this is. Based on our sequencing information we have in the lab, these are the original strains, and they're genetically very, very similar, all these cases. And so we compared, when we started the spike gene sequencing, we looked at 15 PED positive cases, and 10 of those cases, and this is just a small subset of the number of samples we had, but 10 of those cases were almost 100% the same as the original virus that we got in. But then since that time, we've got other cases, and we have these 5 cases in this particular study that are, they're almost identical to each other, 99.6 to 100%, but they're only in this 93 to 95% similar to the spike gene and these others. So that raises the question, is this an additional virus, or is this an evolutionary process that occurred after the virus came to the United States? Our scientists believe that this was most likely an additional introduction from the initial introduction, and they may have come in together because we have some data. When we went back and did the retrospective studies, it looks like that second virus has been around since sometime at least since June. I don't know. I think it's very important to know whether the viruses are still coming in through the portal that the original came, or did they come together which gives us some sense of security, I guess, if it came together. So a little bit on the Delta Corona. You know, it's not PED virus, it's not TG, or it's not respiratory corona. These are the alpha coronas up here, which would include everything except the Delta Corona, and then these are all the Delta Coronas. But field cases, field reports are that this virus causes very similar signs to the PED or TGE, vomiting, diarrhea, explosive diarrhea in baby pigs, suckling pigs. But again, we don't know very much about that virus, and we're struggling to adapt to cell culture growth, although they're working on that. But we can do the sequencing. This is just the positive PED the positive Delta coronavirus cases that we've had at Iowa State. And when we started doing this in February, we went back and did some of the retrospective studies, and so we do have a case from back in December that we know of, and then some of these other cases, as it looks like, again, that's somewhere just like PED, the number of these cases has peaked, but you know, this is not a very large number of cases that have come into the diagnostic lab, and we just don't know, you know, how pathogenic this virus is. And so, and then this slide is just a cumulative positive PED, Delta coronavirus cases by farm type, and from the 22nd of December, most of these cases have been that have been reported have come from sow farm sources. And this is the number of states where we've gotten a Delta coronavirus. It's not a large number. I think there's only there's 73 cases now, Lisa, in our lab, Delta corona. 71 on this chart, and then the states that they've been found in. So.